welcome to the office hour for exploring biological assemblies in the PDB, and I'm Rachel Green, and I will try to give a little introduction and then answer any questions that you might have. So we're also joined by Christine Zardecki, who is the Deputy Director of the RCSB PDB, and we will be recording this office hour. Okay, so just to start, so this is so we're going to do this at kind of a beginner's level, and then if we have more advanced questions, but I really wanted to, we get a lot of, so I run the help desk for the RCSB PDB, and we get a lot of questions from users who aren't quite sure about what biological assemblies are and how they work and how to visualize them. So I'm gonna kind of walk through some of that, and then again, happy to answer any questions. So I'd like to first point you to the PDB 101 training and outreach portal of the RCSB PDB. And in particular, from there, I'd like you to point you to this guide to understanding PDB data, and then finally to this section on biological assemblies. So this is a really great background piece. If anybody I'm going to kind of talk about it a little bit, but then if anybody wants to refer to this afterwards, it's there, and I think it's very helpful. So just to start talking about what is a biological assembly. So it's a little bit of an artifact of X-ray crystallographic structures. So when a researcher does a study of an X-ray crystallographic study, they study what's called the asymmetric unit, and that's the smallest group of molecules in the crystal that gets repeated through the crystal. So the asymmetric unit can be one chain or a number of chains. And then that gets repeated by symmetry in the unit cell. And then sort of just like a three-dimensional brick wall, that unit cell gets repeated through the entire crystal. So when you're looking at an X-ray crystal structure, it's really kind of an averaged picture of all of these different cells. And then depending on the quality of the cell and how the molecules crystallize, you may have fewer or more molecules in the asymmetric unit. So the example that we like to use a lot is hemoglobin, because everybody kind of knows hemoglobin. So I'm going to start with this structure 2HHB, and so every structure in the archive has, at the RCSB PDB, a summary page. And there's lots of information you can get from the summary page about any particular structure. So just to start, let's look at this image and what you can toggle here. So in this case, the asymmetric unit of this molecule, so human deoxyhemoglobin, is the same as the biological assembly. So you can scroll down here and see that there are two macromolecular entities. So I just wanted to talk for just a second about what is an entity. So an entity in any structure is a chemically unique part of the structure. So in this case, there are two copies of this hemoglobin alpha chain, and they're given chain IDs A and C. And then there's two copies of the beta chain, B and D. And all these, so each of those this is an entity, and this is an entity, because those chains have the same sequence. Okay, so making up the asymmetric unit are these four chains. So two copies of the alpha chain and two copies of the beta chain. And this is the same as the biological assembly. So the biological assembly is what we believe to be biologically active. And sometimes that's very clear. Like in this case, we know hemoglobin has all four of these chains. In other cases, which I can show you in a little bit, it's a little less clear, and it's determined by how it appears to be binding, what we know from other experimental evidence. But sometimes the author and the software that we use can disagree. So I'll give you an example about that in a second. So next I want to show you another similar hemoglobin structure. And in this case, the asymmetric unit has only the two chains. So it doesn't have, and you'll see down here, it has one chain of hemoglobin, the alpha chain, and one chain of hemoglobin, the beta chain. So in order to build the biological assembly, here we take two copies of it. So this is a little confusing. So sometimes when you come to this page, if you click on this, you might say, where is the rest of it? Well, this isn't what I know hemoglobin looks like. But if you look here, you'll see the whole thing. And so what does that look like when we try to visualize it? So this is the mole star viewer. And so here it comes right in with the biological assembly with all four chains. If you do want to see the asymmetric unit, you can switch here to the model. So that's what you get here. So you can go back and forth between those. Going back now to the main structure page again, if you want to download these coordinates, you have the choice of downloading either the asymmetric unit, which will give you the two chains, or you can download the biological assembly here. And it does come as a compressed format, which you have to uncompress, but that here will give you all the chains. So I think, so that's a little bit confusing sometimes, that people download this file and expect the whole thing, but what you need to download is this other file. Just to show you also, these downloads are available from this download menu. If you have a number that you'd like to get, you can list them here, and you can select if you would like to have the biological assembly. So you can do that. Just to give you another example of a different kind of situation, this is another that can happen. Here's another hemoglobin structure. And in this case, the asymmetric unit has even more molecules. So it has two copies of the biological assembly. And you can see this again here, lots of chains. So the reason this happens is if there's some kind of asymmetry in the crystal structure, there's some slight differences structurally between the different copies, this will happen. But it's really still the same biological molecule, and so we end up with biological assembly 1 and biological assembly 2. And you can, so they're the same thing, they're just very slightly structurally different. And so again, you can see this if you want to download, you can download either of these assemblies here. And again, they should be pretty similar, but in the case, let me show you another structure. Here, this is the biological, there are two different biological assemblies. So this is the asymmetric unit, it's this. And then in one case, the biological assembly is shown as this, and another case it's shown as the dimer. So why is this? So often software will report one case, and the authors will report another case. And so when the annotators are working on these structures, they'll look at different kinds of evidence, and often they will present both of them as possible viable options. And when you look into the mmsif file, you can see. Let's see if I can. I'm not going to open it because I have to uncompress it. You can see that one copy is determined maybe by the author, and another copy is determined by software. It'll tell you in there. Let's see, what else did I want to show you? Okay, hang on, let me go back to here for a second. So in this, back to this Guide to Understanding PDB Data article, it gives a little example of what does this look like in, sorry, I entered in the wrong place. What does this look like in the mmsif format? So I won't go through it all, but if you kind of want to get into the nitty gritty of how this looks in a coordinate file, you can come here. It'll tell you the transformations that you can use to generate it. Let's see. Oh, I wanted to just okay, then if you want to, before we get to some questions, I wanted to show you a quick search. So here I just did, so I went to do this, I went to advanced search, and then I entered in hemoglobin as a text search. And just to show you, I pulled up this 1HV4 that we looked at just now. And I had it, you can choose if you wanted to report assemblies or structures or macromolecules. So I just wanted to show you, this was the example that I gave where there were two biological assemblies in the asymmetric unit in the structure. And so this shows you, when you do a search for this, you can pull up both these assemblies, but if you search for structures, if you reported structures, it'll just show you the one. And then just another cool structure that I wanted to show you was, just because I think this is pretty cool, there are also some fiber diffraction structures in the archive, and these are built from an asymmetric unit, maybe just of one molecule, and then you can build up a whole fiber with it by using copies of that one molecule.